basics of mushroom identification class for the North Texas Mycological Association. This is the first class we've ever had on mushroom identification, and I'm honored to be able to host this. We've got a sold out class today, which is awesome. Thank you all for supporting mycology in the North Texas region and helping advance it. So basically all the funds that we generated today are going towards purchasing microscopes for the club. So in the future, we can have a microscope course and then people can come in here and learn more about microscopy. So other than that, before I get started, or as I'm getting started, I kind of put a table of contents in here. This presentation is being recorded, so all of you will have time to go back and view this again. So Ben, if you don't mind highlighting that number 2 for the Zoom people. Right there is a little page number, right? So I included a page number on each slide. So if you go back, once we kind of get into this, you're going to see a bunch of different stuff that you haven't seen before, probably, and you want to go back to learn about specific ones. So that's kind of why I added this feature in there. So I'm just going to spend a little bit of time on the first few slides. So whenever you go back, you kind of know, this is the page you look for, right? So first just has the contents on all the pages that I have for the slide, so you can kind of go through and find out. So basically, the PowerPoint starts on slide 19, the mycological glossary. Basically, you're going to hear a lot of terms that you don't really know, and we'll get into that later. The foundation of fungi, understanding ecological roles of fungi, mushroom life cycle, how to start identifying, mushroom anatomy, simple advice to beginners, spore-bearing surfaces, gill attachments, and cap morphology. That's really important. We'll get into that in further detail. How to name a mushroom, 2 phylas, spore print colors, identifying mushrooms with pale spores. And then there's all the white or pale spore genres for the most part. We'll get into chlorophyll. And there's some more pale spore genres that we're going to be talking about. And then the top part is pale spore genres, and then the bottom part, we're getting into pink spores, and then we'll kind of talk about that later on. So then more into brown spores. So basically, the spore coloration is very important in mushroom identification, especially for beginners. We're going to be talking about this in detail in the presentation. Oh, I can't. We're going to be talking about this in detail pretty soon. So these are all the dark spore genres that I'm covering today. The Boletaceae is mushrooms that have pores instead of gills. So we'll be talking about those too. And then polypores, the ones that we find on wood mostly. And then we got kind of the morels, and then we got the regular different kind of genres that are kind of miscellaneous. So we're going to be talking about those as well. And that's kind of like the end right there. So this kind of long presentation. If anybody has questions, I'll try to cover it real fast. As you see, I've got a lot packed into this. So we got to kind of move forward. But, I can't wait till the end to take all the questions, because I'm pretty sure you guys will have a lot of questions at the end. So I'm just going to kind of cover them as we go. So basically, mycologists have a specific language they use to describe all aspects of fungi, and it's not Latin. It's called mycospeak, right? So basically, you're going to be seeing a lot of words that are unique to the world of mycology and not applied anywhere else in this presentation. So understanding mycospeak will take time and practice, just as learning any language does. There's some good references right here at the bottom, which is Mushroom Expert. And then also, Dr. Rob Halleck is a good friend of mine on Facebook. He created this book, and it's really helpful. He also has this group called Mycological Word of the Day on Facebook. So if you're interested in all things regarding Latin mycology, then you should join that group. The fungi kingdom consists of single and multicellular organisms that play an important role in the ecosystem. There are very few unicellular fungi, and mainly they're the yeasts. So today I'll just be focusing on the multicellular fungi that we're all familiar with. Multicellular fungi are formed when spores, the reproductive cells of fungi, find their preferred habitat, and under the right conditions, create thread-like cells called hyphae. Hyphae then will begin to branch and fuse together to form mycelium, the foundation of all the fungi I'll be discussing today. So once the mycelium builds up, it forms what's called a primordia, and it's kind of like a little blob. And that little blob kind of turns into the fruit body you see when you find a mushroom. So that's the initial stages of a mushroom. And fungi are among the most widely distributed organisms on Earth. They outnumber plants 6 to 1. Some estimates claim there's over 5 million species worldwide, most of them yet to be discovered and identified. So fungi have 4 major ecological roles. Saprobic being the main one, which we find a lot here in North Texas. They're known as the decomposers of the organic world. So basically, whenever you find a mushroom going on like a little piece of a stick, it's probably a saprobic fungi. It's just decomposing that organic material. And then there's also mycorrhizal fungi that are formed when fungi share a symbiotic or mutually beneficial relationship with the roots of certain plants and trees. Mycorrhizal fungi permit the host plant to absorb additional moisture and nutrients, and in return, the fungi receives polysaccharides or the carbohydrates necessary to survive. Pathogenic and parasitic are 2 very similar, except pathogenic infect the living plants and organisms with the sole purpose to cause a disease, right? So both of pathogenic and parasitic fungi infect living hosts. But the difference between pathogenic and parasitic is pathogenic does it with the purpose to cause a disease. So that's pretty much the 2 differences right there. So this is the mushroom life cycle. Basically, if you can kind of follow my arrow. Okay, so right here is the germination. Whenever the spores, they are the foundation to all mushrooms, right? So if you know plants, think of these as the seeds of the mushroom world, right? So these are under the right conditions, whenever it's like really moist, has a bunch of rain, and temperatures are right, there's not a lot of drastic change in temperatures, fungi will then find the right conditions to create hyphae and then develop into what's called the mycelial expansion, which is right here. And that's whenever the hyphae kind of fuses together. And then that's when you see a bunch of white strands, and they're really cottony and wispy. And usually after that, once the strands build up enough, they kind of form into a primordia, which is a little blob that is trying to sprout up and is just beginning its life cycle. As soon as it starts to build a formation, it starts to get into a little bit of a recognizable stature that we see today. And then once it's mature, it starts to release its spores again, and then it starts the life cycle all over again. So that's the mushroom life cycle right there. Okay, so how to start identifying, right? Basically, you're gonna need to know a few things before you can even start to identify mushrooms. The most important is the habitat, right? So if it's growing on wood, then you can eliminate a lot of suspects like mycorrhizal fungi, right? Because mycorrhizal are specific to growing off of the roots on living trees, right? So, you can kind of eliminate a lot of genre just by understanding the habitat, right? So if it's saprobic, if it's growing on wood, if it's the wood dead or alive, these are all really important factors to include whenever you're writing a description or trying to identify a mushroom. And then also, especially for beginners, the spore coloration is very important. As I said earlier, we're going to get into this in detail, but this allows you to eliminate a bunch of genre, right? So if you have a mushroom with dark spores, then you can eliminate all of the white spore genre of mushrooms, right? So it's process of elimination, and then you kind of get to your answer there. So the gill attachment is very important as well. We're going to get into this further detail in the next few slides. The shape and color of the fruit body, odor. Some fungi, they really stink, or they have specific odors that, helps you in identifying. And also taste tests, so as opposed to plants, fungi, you can perfectly, it's safe to nibble a little bit to test for an accurate taste or peppery, or sometimes it's just really bitter, you know, and that kind of helps you in identification. And so most times at all, you won't, this is just kind of bland, you know, but these are all factors that kind of help you in identifying. Also the location, right? So location is a very important factor. I'll give you one good example. It's the Texas star, right? It's our mushroom, our state mushroom of Texas. And basically it's only been found in Oklahoma, I think a couple times, but we'll let them have a couple of instances. But anyway, if you're looking for the Texas star and you're in the East Coast or the West Coast, you're never going to find it, right? Because it only goes in Texas. So location is very important, and kind of writing down where you found your mushroom helps us identify it. And also the ecological roles, as I kind of highlighted earlier, you know, if you found it next to a tree, you want to make sure, is this tree a hardwood or is it coniferous, right? Does it hold onto its leaves throughout the year, or does the leaves fall off, right? So those are the two big identifying factors. And any change in color once pressure is applied on the fruit body. And we'll get into this in more detail here a little bit. So the mushroom anatomy, you know, so we're gonna go from the top down right here. So this is kind of a weird diagram because it has a bunch of different mushrooms fused into it, right? So don't look at this and think it's like, you know, the mushroom to look for. So basically what I'm pointing out here is this right here, for the most part, has the look of an amanita, right? It's a very robust specimen. But right here, can you kind of zoom in? I'm not sure if yeah, there you go. Yeah, so right here, if you can see, it has a little bit of a pore surface. And then right here is the gills, right? So the spores are kind of falling out. Areolae is the cracking surface of whenever the Peleus tends to crack in certain formations. The zonations, you'll see concentric lines whenever on a lot of different species. So you'll have to notice that as well and put it in your description. But if you have a picture, you know, so we can kind of see that in the pictures and kind of identify it from there. So the scales, usually those are very common with Amanita species, you know, and they're removable as well. Kind of the wart, oops, sorry, kind of messed you up there. The warts are very similar in Amanita. So the scales are different because you can't really remove them from the cap, as opposed to the warts, right? So in Amanita, you can remove the warts. They're part of the egg that we'll kind of talk about in a little bit. And then the tubes, right? So tubes are whenever you break a polypore or something that has pores, and you look on the inside, you'll see a bunch of tubes, and we'll get into that here in a little bit. But that's a good indication that you found a polypore or something that has pores, especially if it's really dilapidated and you really can't tell on the outer surface. And then sometimes you'll have teeth. You really can't tell from this diagram here, but they have spine-like projections coming down from wherever the spores are dropping. And right here is the skirt or the ring, or sometimes we call it the annulus in myco speak. The reticulum is kind of not really well defined here. It's basically, it's a fishnet pattern found on Boletaceae, the ones that have pores on the undersurface instead of gills. So they have this regular stem, and then the stem has a beautiful fishnet pattern on some species. And then the amount of reticulum is very important factor as well. And then on all Amanita species, they'll start their life cycle in a little vulva or egg and sprout out whenever it starts to reach maturity. Also, there's scales on some species and genre. You really can't see it, and it's not really that well defined here. And then the mycelial roots right here, and then there's the basal bulb at the end. So let's see, let me try to zoom back. Okay, thank you. Oh, good.
There we go. Sorry.
Yeah. Yeah.
Okay.
Okay. So a few more tips I just kind of want to cover. You want to check the spacing of the gills, right? Because sometimes they're really close together. Sometimes they're spaced further apart. That's very important. And then the pore size, is the pore angular? You know, the shape of it's really important as well. And then the reticulum or reticulation is the netted pattern I kind of just covered. So simple advice to beginners, you want to spore print it. If you find a mushroom with gills, it also works with other genre, but mainly with the gill fungi, you'll be able to eliminate a lot of genre and kind of go forward from there. Do you want to take the cap off and place it on some foil for about an hour? That gives it enough time to drop the spores and give you a spore print coloration. And then also I kind of covered what is it growing on. This is very important because, you know, certain mushrooms that just grow on dung or on dead wood or after a fire has been, you know, through the area, you know, so all these are very important in identification. And, you know, you'll see this later on. You want to carefully excavate the fungus, right? If it's growing from the ground, because, and I've done this plenty of times, and I've even included an example to kind of show you guys where I broke the base when I was trying to excavate a mushroom. And there, as you can see here, this one has what's called a rooted stipe. That means the stem digs really deep into the soil. And it's a very telltale characteristic of this species right here. So basically you want to make sure you get the entire specimen whenever you extract it.
So will the spore stick to the foil?
So a member asked if spores will stick to the foil. Usually people will take a bowl and then they'll kind of flip it upside down, you know, to give it a little bit, but generally people do it in their house, you know, so the spores do stick to the foil and you'll get a beautiful little spore print. So you can kind of keep in, you know, show to everybody else. So this page right here is very important. It covers a lot of different features to check for when you're looking for an identification. So there's several types of spore-bearing surfaces, also called hymenophores. They include gills, pores, ridges, or teeth. Gills, pores, ridges, or teeth. We're going to zoom into this as soon as I'm done covering this. And many of these are found in the phyla Mycota, and we're going to get into that pretty soon. The connection between the hymenophore and the stem is probably one of the most important features to check for when it comes to identifying a genus. In most cases, it will be the gill attachment, but it also helps in identifying certain species in the family Boletaceae. Usually an undershot is recommended for this purpose specifically. So I'm not sure if you guys try to post your mushrooms on Facebook, and a lot of times you'll get chastised if you don't have an undershot with them, right? Because people like me, we get annoyed, just, you know, we want you to get good photos of a mushroom up close, because people take a photo of a mushroom from 10 feet away and expect us to identify it. And I'm sorry, we just don't do magic. So basically get up close photos and pretty much pick up as much details as you can, because all these gill attachments that you see right here are very important in figuring out which species you probably have or which genus you even have. Because a lot of times you probably won't even make it 2 species. So just if you can identify 2 genus, it can kind of figure it out from there, hopefully. The cap or pileus of a mushroom will usually have various distinct morphological features throughout its life cycle. The color of the cap is liable to change as the specimen matures, a term mycologists called hygrophanous. Once the cap matures, it opens up and usually develops a different shape or goes through a series of different shapes. These are all very important characteristics to check for in the identification process. So Ben, if you can kind of zoom in. Yeah, there we go. So basically, oh, sorry, I kind of messed you up. Sorry. Sorry. Okay, so gills we're all familiar with. This is the pores that I was talking about that are familiar with Boletaceae. And then ridges, right? These are what you see usually in some Cantharellus species, the chanterelles, and then teeth, right? So these are all spore-bearing surfaces, also called hymenophores. And we don't really get that many fungi that have teeth formations growing right out of the ground in North Texas. So, you know, but it's important to understand that that's a feature to check for as well. So, and then also the gill attachment, right? Is it adnate? Are the gills widely attached to the stem? Are they narrowly attached to the stem? Are they running down the stem for decurrent? And then emarginate is gills notched immediately before attaching to the stem. So these are all really confusing. And even me, years after, I still have to refer to this chart. So I'm just telling you guys, you're gonna have. I think Ava had a printout that each one of you guys hopefully grabbed. And you're gonna. Did you have a question, ma'am? I don't, but it would be really useful too. Yes, yeah, just so you can get a kind of a grasp on all this information, because what I'm covering today, it'll take a long time for you to just digest all of this information. So yes, and then if you have the actual copy, maybe we can reach out to the Zoom people and give them the copy as well. So as I said, these are very confusing. Some of the most important ones I'll highlight right now is free, right? That means the gills aren't attached to the stem, right? So when the stem comes up, it'll have a ring right before the gills start to appear, right? And that ring area is what we call the free zone, right? So it's not really attached because all the attached fungi have gills that come up straight to the stem. So that's the main difference there. And then decurrent also is important. You'll see this on a lot of different mushrooms where they're running right down the stem. And they're totally completely different from free. So free is not connected to the stem, and decurrent is running down the stem, right? It's kind of going down the stem. So, and then also the cap morphology is very important as well. You know, a lot of these you might recognize just by looking at it, like the ovate shape. That's kind of like a regular mushroom. And then a lot of times after mushrooms grow and age, they have like a flat top, you know, when their gills spread out. And a lot of times they'll just remain one specific shape throughout their entire life cycle, like the conicals, the conical shapes, you know, they really love to have that little umbo, is what it's called, a little nipple, so to say, on top of the Philius as defined right here on top. As you see, that's what the umbo is. So now we'll just go on to the next slide. Okay, so this was the example I was telling you about that I kind of messed up the base there. This is a deadly mushroom called Amanita ameriverosa, and identifying them from the base is very important, but as soon as I kind of plucked it, I kind of the base kind of snapped in half. So that's something that you don't want to do. As you see right here, the base is broken. And as you see, there's very small remnants of a vulva coming out. And that's what the Amanita starts their life cycle in, is a little egg connected to the roots of the tree. And then it starts to pop out of that egg. So how to name a mushroom? Usually you only see a mention of the genus and species, and sometimes only a genus, you know, if people can't get to the species. But the actual full written out text is right here at the bottom, and it's entirely way too long for us to write all this out, and it's very confusing to say the least. So that's why we go with the genus name, which is always capitalized, and then the species name at the bottom, which is always lowercase. So whenever you every single name besides the species will be capitalized. So if you notice right here, it's from the phylum Basidiomycota, the class Agaricomycetes. Agaricales is the order. Amanitaceae is the family. Amanita is the genus, and Ameriverosa is the species. So this is way too long to write all this out. So that's why we just go with Amanita Ameriverosa, and that's the accepted terminology. And then also, I didn't really cover this, but you'll see a lot of common names being used. And I really don't encourage common name usage because it varies from different region to region. Sure, in North Texas, we might understand what you're talking about, but if you're talking to somebody from California, they'd be like, what is that? You know, like, so common names not really always accepted. That's why Latin is universally accepted, and that's why we use it. So two phyla you should know are Ascomycota and Basidiomycota. And the main difference between these two is the way they produce their spores. Okay, so Basidiomycota or Ascomycota have spores inside an ascus right here. And as you can see, these are the spores, and the outer layer that's kind of holding it is called the ascus. And that's where it gets the name Ascomycota. And members within this phyla are Ascomycetes, and this is the largest phylum of fungi with an estimate of 65,000 species identified. And usually these are the ones that you find the crust fungi growing kind of flat on wood, or you know, morels might be something that you're more familiar with, because in the next one that I'm covering is called Basidiomycota. That's mainly the guild fungi, right? Because Basidiomycota, they take their spores and they release it from the edge of the gill surfaces. Okay, so the spores are attached to the gill surface for most Basidiomycota, and that's how they release their spores, as compared to the ascomycetes, where they have their spores inside what's called an ascus. Okay, so that's it just shoots out. Yeah. So and then oh, I'm sorry, a member asked how they release those. It just shoots out. So basically, Basidiomycetes are what we're most familiar with, the guild fungi. But they do include some other genres, from polypores to stinkhorns to jelly fungi, tooth fungi, coral fungi, and more. They're kind of very diverse, and it's very confusing because um, the Basidiomycota right here, where it has the gill faces, you don't really have a gill on a stinkhorn or a polypore or any of these other categories that I've covered right here. So in those cases, it just kind of releases from wherever the spores are created from. The stinkhorns is different because it has their spores in the gleba, which I'll cover later on. And polypores, they just kind of release it from where they're at. So moving on, we're going to go into the spore colorations. So this will kind of give you an example of your reference to your different colors that you should expect to see when you're spore printing mushrooms. And the pale spores could kind of range in coloration. I've seen them anywhere from completely white to kind of a beige to a little bit of a rosy pink. And we kind of still categorize the rosy pink under the pale spores because it's not a true salmon pink coloration as this one here. So there's two main families, the Entolomataceae and Plutaceae. So we're going to get into those in a little bit. And then the dark spores are all the ones that are not brown and rusty orange coloration. So we'll get into those towards the end of the presentation. We're starting off with the white spores, and then we're going to just go into this order. But mainly in Texas, we get a lot of the white spores and we get a lot of dark spores. So these are the two main ones that you should focus on. And sometimes we get a little bit of the orange one, or brown, sorry, brown one. So identifying mushrooms with pale spores. Pale spores include colors that range between white to yellow. Rarely you'll have a hint of pink, as I just mentioned. It's not salmon pink. So there are many different genres that fall under this category, and they're probably some of the most often encountered. So expect to see a lot of white spored mushrooms whenever you're out there. And today I'll be focusing on some of the most common genres that we encounter whenever we're going out into the woods. And for the most part, unless I know otherwise, most of these are found growing on the ground, okay? So Amanita, most of you probably recognize, this one's pretty robust, large specimens anywhere. They're rarely very small. They're mostly medium to large in size. And typically, as you can see at the bottom of this picture on the top, you see this little egg. It's called the vulva, right? So this was the vulva or egg-like formation that I was telling you guys about, that all Amanitas start their life cycle in. And typically this vulva can be buried underneath the wood, underneath the soil, and or it could not even be present, or whenever you're trying to pull it out, it might not even have enough membranous tissue remaining for you to even identify it as a vulva. So those are all telltale characteristics to check for and include in your description. Hey, when I was pulling this out, I had like this little membranous tissue, that came with the mushroom, and that's the vulva. So make sure you kind of excavate around the mushroom to make sure that you don't lose any of the details. And so they're all mycorrhizal. They grow right next to specific trees that they kind of share a symbiotic relationship with. And they grow right off of the root system, and they provide those roots with water and in turn receive their nutrients to grow. So often you'll see removable scales on the pileus or the cap. These remnants are known as the universal veil left over after the amanita protrudes out of the egg. So at the bottom of this picture right here, you see these beautiful golden flakes on top. Those are warts that I discussed earlier, right? And you can kind of remove them as opposed to scales, which are, yes, ma'am, do you have a question? Oh, I'm sorry. As opposed to scales, which are fixated on the cap. They're always dry and never hygroscopic, which means that the cap never changes color as it matures, which is a very important characteristic to kind of tell for, look for. And then the base of the specimen is very important to kind of cover. And it's broken up into 7 sections. Vaginate, it doesn't have an annulus, right? So this one is kind of a bad example because it does have an annulus, but it's kind of hiding right there, right? So in younger specimens, you might not be able to recognize, you know, if it even has a veil or not. But usually you should be able to look right there. And as you can see, it kind of has like a little bit of tissue hanging off, what we call appendiculate, right? So it's kind of like a skirt hanging off the side of the margin. So that one you can kind of tell it had an annulus as it's trying to open up, right? So, and then, so the vaginate, so basically, this isn't all the specific details. I just try to cover some important ones because, you know, I can't really cover everything. So in Europe, we often cut mushrooms to preserve roots. Is there a reason we excavate versus cut? Yes, you know, and then I've heard this this kind of be brought up quite often with morels. People say to, you know, cut the morel and leave the stem base and the actual mycelial mass in the ground to kind of allow for reproduction purposes. But there hasn't been any concrete proof that this actually helps in any beneficial way to preserve the specimen. You know, so basically for Amanita, you don't want to cut off the base because you're missing a lot of the details, right? Because with Amanitas, especially that specific base, if it has concentric lines, as this one does at the bottom, it's kind of hard to see. And then this one's kind of just covered entirely by the vulva, right? So if you cut it off, then you won't really get any of these details. So, and then section Caesare is the edible ones, right? This one is in section Caesare. The base stem is cylindrical. So it's kind of hard to tell when it's got a vulva like that. And so the section Phalloideae, I actually brought a specimen here, is the deadly ones that we're kind of familiar with. So they always have a vulva and they always have an annulus, but they tend to be white in coloration. So if you're not really familiar with mushroom identification, don't really go out and eat white mushrooms. Okay, so okay. Anyway, section Lepidella, the margin is decorated with velar material that is more persistent. So as opposed to this has kind of got velar material all over the cap. Section Lepidella would have it all over the cap, and then it'll be hanging off the margin as well. You know, so Lepidella and Amidella are kind of very similar, but the velar material on the margin disappears sooner with Amidella. And then Validae is another section that has a bulbous stem that's kind of really abrupt, and it's less defined than section Amanita, which is this one right here. Section Amanita usually has warts on the cap, as you see right here, in concentric circles at the base. So that's really main identifying characters within Amanita. Can you? Okay. So Rusula and Lactarius. These two are very similar when you look at them. Their statures, and that's why I kind of include them together. They're both mycorrhizal, which means they grow off of the ground attached to the roots of certain trees, just how Amanita does. But main differences between these, a lot of species in Lactarius exude a latex whenever you cut it, or even just as soon as you kind of pick it and look underneath, it just might be exuding latex right then and there. And a lot of people, including myself, my first thing I do is lick the latex. And a funny story is I was walking in this park and this lady was I didn't know this lady was looking at me, and I was just pinned down I pick up his Lactarius and I'm like, ooh, and I just start licking it. And I'm looking over and this lady thinks I'm crazy. So basically, you lick the latex off the Lactarius to test for bitterness, right, or spicy flavor. As opposed to biting into the mushroom, you can kind of get a glimpse of it then and there. And Lactarius, I'll tell you, I've bitten to a few that I wish I wouldn't have. So basically, taste test might be a very important identifying characteristic in Lactarius. So the russulas, they're very recognizable. They're brittle. They're kind of known. Whenever you throw them against the wall, they bust apart like chalk. Maybe we'll do that one day in the woods, so it disperses their spores or something. And you know, they tend to have a white stem, right? They can usually be different colors on the cap. Sometimes they're very beautiful colors. They can have green. There's a lot of red ones. In North America, we have like 60 to 100 red cap species that look just like this. So whenever you do find one and you try to ask for identification and you want to get to species, we just tell you to go look at it under a microscope, because even then it'll be difficult to identify because there's 60 different ones that you have to go through and eliminate. So russulas, they also have a unique characteristic to where you can peel the skin off of the cap, right? And whether it peels entirely or moderately or barely none at all, that's all different characteristics that you would kind of make note of when you're making an identification. And then also, no, this one's attached just by a notch, so I'd call that add next. Yeah. So and then this one's just kind of the same. As you can see, it's barely running into the stem. When they're free, they'll have like a ring around the base area, and then it's a little bit of a space in between the gill area and the stem. But what's russulas, the spore color can range between white to yellow to pale orange, which is also a helpful indicator. And a lot of times you won't even get to species with russulas because they're that abundant, that common, and that boring. People don't really get that much into russulas because there's literally 1,000 of them. And Lactarius, this one's a little bit easier to get to species because the latex is a lot of the key in Lactarius, right? So it can sometimes bruise different colorations also whenever you slice them, and it could cause the actual specimen to turn a different color as well. So all those are important characteristics to check for when making an identification. And usually if you see on the edge right here, the russulas, they kind of, their margin, they kind of open up, right? And then Lactarius, they're kind of known for having their margins enrolled, so it's kind of curved inwards. So that kind of goes towards the cap morphology that I was covering earlier. And then basically, you know, you'll also see concentric circles on the cap, which is a very common feature. I should have kind of included it here, but sorry, maybe next time.
That's on the top of the cap.
Yeah, on the top of the cap. So whenever you flip it over, it'll usually have some zonations, as they're called, on the pileus. Okay. Oh, yeah. So the waxy caps, the hygrophoracea. So waxy caps have a glossy sheen to them, right? They kind of look like they're made of wax, and I've even heard they taste like wax, but I haven't eaten them. So there's no toxic waxy cap. So you know, I guess take that for what it's worth if you want to go try to eat one. But make sure that you have a waxy cap before you start eating one. You know, always ask for identification before you eat something. And waxy caps are just loose terminology applied to members within the family Hygrophoracea. These species tend to have bright, flamboyant colorations with a glossy sheen to them. Members within the genus Hygrocebi have uncertain ecological roles, with recent studies showing many of them are mycorrhizal or parasitic on different types of grass. It's really cool, you know, because I don't really see a lot of mushrooms being parasitic on grass specifically, because up until, you know, I read about this, whenever I would find a mushroom growing out in the open field where there's like no trees or anything nearby, I would just assume that it was saprobic decomposing the grass right there, right? Because there's no trees, there's no buried wood visible, so the process of elimination would tell me that it would be saprobic, but come to find out, some of them have a symbiotic relationship with certain grasses. And then also, usually they're very slimy whenever you touch them. That's another telltale characteristic. It's called viscid, the terminology for that. And then their gill spacing, as you can see right here, is a little bit further apart than, if I go back, you see how these are very close together, and there's so many of them as compared to the distance between these gills and the amount of actual gill faces that are present. So those are all very important characteristics to kind of look for. Lipiota and Leucocopernis. So these can sometimes be confused with Amanita, right? And the important characteristic between these is they don't have a vulva, which is the egg that Amanitas start off their life cycle with. The thing about that is some Amanitas, like I mentioned earlier, you won't be able to recognize the vulva as present, right? So it can kind of be tricky. But when you look at these, a lot of them have scales, right? So this would be like the cracking feature that I was kind of suggesting earlier. Whenever the cap opens up, and usually they're small like this, but whenever it opens up, it starts to cause little cracks on the cap itself. And Amanitas don't have this feature, so that's a good characteristic to kind of look for. So basically, in this group, I'll be discussing these species will look similar to genus Amanita. They're all similar in stature, and some of these genres, they possess an annulus or ring around the stems. They also have free gills, which means that they're not attached to the stem. Amanita also has free gills, so that's where it's really confusing. And then Lipiota, right? So Lipiota, I kind of should have named these, but Lipiota is this one right here, and they're very similar to each other. And even now, you'll see mycologists or people on Facebook when they're trying to identify things, they'll get these mixed up together because the identifiable characteristics are very similar whenever you're trying to get to the actual genus. So Leucocopernis is more tropical. It tends to be smaller in stature and extremely delicate with a well-developed annulus that usually it does disappear over time. Like this one here, it was developed, but now it's kind of slowly disappearing. And this one's kind of disappeared completely. And then as you can see, it has small little granular scales, which kind of looks like sand or little particles on the cap. And those particles are also removable. So whenever you touch them, it'll stick to your finger, and that's also a good indicator for Leucocopernis. And then these are also Chlorophyllum, Macrolipiota, and Leucoagaricus. So all these are very similar to Amanitas, right, in their stature. They can be the same size, but all these are saprobic as compared to Amanita. They grow right next to trees. And with the exception of Chlorophyllum molybdites, which is this one right here, all these have white spores. And the only reason I kind of include this one here is because it's in Chlorophyllum, but it's the only genus found in North Texas that has green spores. Very common. You'll see it here in spring whenever the rains occur. And it loves St. Augustine. You probably get it on your front lawn at your house. And it's responsible for some of the most, probably the most reported toxic cases of mushroom digestion in North America, because people see this growing out in their front lawn and they're like, oh, wow, this looks like the store-bought mushroom. So they go out and they eat it, and then they start puking and explosive diarrhea and stuff. Yeah, so we don't want to eat that. So if it's green spores, stay away from it. Okay, that's a good rule to remember there. And then so Macrolipiota is this one right here. As you can see, it has. Can you zoom into that one, Ben? So this one, it has a distinct tiger stripe pattern on the stem, right? So that one's a little bit easier to separate from the others because of its distinct pattern, and a lot of the species has this. So basically, that's what you want to look for when you're identifying Macrolipiota, and you're trying to separate it from these other genres that I just talked about. All right, can you kind of zoom back up? Yeah. So next one is Leucoagaricus. So I have one of these specimens here too. This one at the bottom left is Leucoagaricus leucophytes. This one is commonly mistaken for the deadly Amanita I just covered, right? Because when you look at it, it has the annulus, right? And it's white in stature, but you know, you look at the base and you really can't tell because remember that first one that I broke the base off of? You can barely tell there's a vulva there. So the thing with this species is they usually on the exterior, when you go to pick it, it'll bruise brown on the exterior. So on the stem, and that's a feature that you don't see in the deadly Amanitas. And the deadly Amanitas, they don't bruise at all. So they'll just remain white throughout their life cycle. And this one's Leucoagaricus americanus. It's very common here in garden landscapes or mulch or, you know, anywhere that, you know, it's kind of like campus setting. I've seen it plenty of times. So you'll probably recognize this whenever it pops up in the spring. And Lacaria. Lacaria is another mycorrhizal genus that is commonly associated with pine or coniferous trees. We don't really get it that often up here in North Texas, but down in the south and southeast and towards the east where there's the pines towards East Texas. Every time I go out there, I find this. So I wanted to kind of cover it. They always have similar colorations, except for a couple notable species that are really purple in color. But for the most part here in North Texas, we find them, they're kind of drab orange, or they have somewhat of a pinkish, rosy pink to, I would say, brownish coloration. And the important factor with a lot of Lacaria is you can't really see, but this, can you zoom in right here? Ben, I'm sorry to keep bothering you. So if you see the base right here, this is the base of the mushroom. And right here is what mycologists call basal mycelium, right? This is what's the mycelium that's left over at the base of a mushroom. And the color of that basal mycelium is very important in certain species or genus, and when you're trying to identify that. And for Lacaria, they always have a white basal mycelium feature, right? So whenever you see like a drab orange coloration of mushroom, right, and you're next to a pine and it has like a, you know, a little bit of white basal mycelium right there, chances are it might be a Lacaria, right? So if you can zoom back up, they have tough fibrous stems, right? And that's another good feature to look for when you identify these. You know, there's not any toxic ones. I've heard people eating them, but stem is so tough that I wouldn't really think that it would be palatable, but I guess, you know, it is in some places. And so that's Lacaria right there. And then we're going off to Mycena, which are usually small mushrooms. They're saprobic in nature and they decompose organic matter. A lot of times we'll find them growing on wood or, you know, they'll just sprout out of the ground. And when I say wood, that's dead wood, right? Because it's saprobic. It doesn't grow on alive trees. So this genus can be found growing on stumps, moss, leaves, pine cones, whatever. They never have a partial veil or annulus, and the stems are somewhat fragile. Species are rather small in stature and somewhat closely spaced gills. Just depending, you know, it really depends, because I really shouldn't have put that there. Some species I've seen, they're really exotic and they have really far space scales, but none in Texas. So I don't want to get you guys too confused. I know you're already absorbing a lot of information. Some are odoriferous, which means they have a particular odor to them, right? Some can be really rancid and some can smell like garlic, you know, and some even bleed when you break the stem, kind of like Lactarius. And that bleeding feature is a really telltale characteristic. We call them the bleeding mycenas, right? Because whenever you break it, it starts to exude latex. So the gill attachment for mycenas can be variable, so it could be kind of confusing there. Marasmias, these are really cool mushrooms. Some of the most pretty ones that I think, you know, are found in the gilled world of fungi. Basically, these are, notice this umbrella-like pattern, right, on the cap. And a lot of times when I see this umbrella pattern, I can almost immediately recognize that it's a Marasmias, right? Because A lot of these species have this kind of riveted pattern where it kind of grooves in and out, kind of like you're opening up an umbrella. And then the gill spacing is a little bit further apart than most other mushrooms. And like I said earlier, you always want to make sure as the gills close, are they further apart? That's really important in mushroom ID. And they're saprobic as well. So you can find these growing on a host of different substrates, right? But never on anything that's alive. So, these can also have a garlic or rancid odor as well. Colibia and Gymnopus, they're very closely similar, and I think they're recently combined into Gymnopus, Marasmiellus, and Calibiopsis. Right. So, and then you'll see this later on in the presentation again, where they'll take a former genus, scratch it entirely, and then put it into a different genre. Right. So, and that's due to DNA sequencing and advancements that we've recently had in the past decade or two. We're really getting to understand where these mushrooms fall, because we've basically been putting them under their macroscopic features up until this point, and under their microscopic features as well. But DNA, you can't argue with that, right? So whenever the mycologists say we're going to change it into this genus, that's what happens, and we just blindly follow them. So they are saprobic in nature and can grow on dead wood, leaves, duffs, etc. Just like the other two genera I just covered. They're usually small to medium in size and closely spaced gills. So if you can kind of zoom in, Ben, kind of check out those gills. Yeah, either one. So if you look at these gills, they're kind of closer together, right? They're really scrunched in together. And like I said, whenever you look at the gill spacing, you want to make sure, is it close? Is it distant? Is it far apart? These are all telltale characteristics to look for. Let me see if I can, yeah, there we go. And then let's see, they usually have a tough stem, kind of like Lacaria. I've took a stem before and wrapped it around my finger, right? So that kind of gives you an idea of its malleable nature. And the photo on the bottom right is Marasmiellus luxurians, formerly in Gymnopus. And you see how confusing things get, right? So you're going to get more confused later on. Yeah, Clitosobi. Clitosobi is a mycorrhizal genus that contains a lot of species that are commonly encountered all over the world. I kind of volunteer for a Facebook group that does poison identification, and this is one of the most commonly encountered species that dogs will eat and start having bad symptoms. Usually they start to vomit, they'll have teary eyes, and then explosive diarrhea, all that horrible stuff. So Clitosobi contains, most of the species contain a toxin called muscarine, and that toxin does exactly what I just said. You don't really want to eat these. And they kind of usually all have a funnel shape to them, right? If you kind of look at it, it's called infundibulicibi. So that funnel shape, it kind of is almost a good feature to look for. And they always have kind of decurrent gills running down the stem, right? Whenever we talk about decurrent, how it's kind of going all the way down the stem here. And so the odor is very important as well. And a lot of these species, they'll have the same coloration. It's usually white, kind of gray, light gray, or tan, like light tan in coloration. So pretty much you don't want to eat these. They're mycorrhizal in nature and grow under certain trees. In North Texas, they really love oak. So you'll see a lot of these in the future.
What do they smell like?
They can have anywhere from pleasant odors to horrible rancid odors. So the pleasant odors is what kind of is responsible for people eating them, right? They're like, oh my God, this mushroom smells good. It must be safe to eat, for some reason, I guess. But anyway, Tricholoma is a mycorrhizal genus that contains a lot of species which are also commonly encountered. They're medium to large as well. And I'm not sure if some of you have heard of Matsutakis. Matsutakis are tricholomas, and unfortunately we don't get them in North Texas, but yeah, they're very highly prized. And I think someone mentioned like $1,000 a pound in some places or something like that. Yeah. So their gill attachment is always attached by a notch, right? So I think this gentleman had asked me a question earlier about that rusula that was barely attached by a notch. And can we zoom in, kind of show the gill attachment to the one on the right? That'd probably be a better one. So this one's kind of tricky because it has a little bit of an area right there. Has a little bit of an area right there that would kind of mistake people for having a ring, but it's really not. As you can see, the stem kind of turns white almost towards the apex right there, and the gills barely run into it. So at times it can be really confusing looking at gill attachments, and you're going to have to look at it over and over again to really understand the terminology and actual apply it in real life. So the tricholomas, they can have a ring around the stem or an annulus, or they can just be bare as well. And usually they really love pine and coniferous trees. So we don't really have that much up here in North Texas, but towards East Texas, there's a lot more. Armillaria and Omphallotus. So these two are very similar, especially I kind of put it in Armillaria, but recently it was changed to Disarmillaria caspitosa. And that's kind of where the confusion comes in, right? So you had one thing that everybody in the world calls the ringless honey mushroom, or Armillaria tabescens, which is this specimen to the left here. And its toxic cousin, Omphallotus alludens, which we have here quite commonly as well. So basically, the difference between these two, if you see, this has a lot more bright orange coloration to it. And this one has scales on the younger specimens on the cap. If you see right here, it's kind of fuzzy, furry, tomentose features, what mycologists call it. And usually that feature will dissipate as the specimen matures, so it's not really 100% throughout its life cycle. But Omphallotus, on the other hand, kind of has a bare surface. Sometimes it has like depressed fibrils, which mean another species, subaludens. But for the most part, you really don't have that feature with Omphallotus. And this one's called a jack-o'-lantern for its characteristic of having a bioluminescent nature, right? So at night, it glows in the dark, which is really cool. One of the only ones that we have here in North Texas that is bioluminescent. So the honey mushrooms is the everything in Armillaria, right? So everything in Armillaria can be eaten. In North Texas, we get a lot of the ringless honey mushrooms, which is Armillaria tabescens, which recently got moved into Disarmillaria caspitosa. They usually go in cespitose clumps. That means whenever they're really bunched in together and they're fused, as you can see with this one right here, right? Usually all Armillarias are edible, but this is the poisonous lookalike, and they're more orange and have small scales. So Pleurotus, aka the oysters, right? So this is like some of you guys are probably familiar with this one. We have 3 species here in North Texas that we see throughout the year. And basically, they each can be separated by distinct morphological features, right? So Ostreatus is the regular oyster that we all know, right? Because of its features, it kind of looks like an oyster, right? They really have what's called a pseudostipe, or somewhat of a stem, right? It's kind of starting to form, but it doesn't really form. So the stem isn't really defined in Ostreatus. But Pulmonarius, on the other hand, as you can see with this specimen here, those are the stems. And usually they're longer and more well-defined. And for some reason, they call it the summer oyster, but I find this all year in Texas because we have freak weather, so, and then Pleurotus laevis, this is kind of not really the best example, but I kind of wanted to show you guys what it looks like in its mature stage, right? Because it's a lot easier to identify in its younger stage because it has a veil or an annulus that soon disappears, right? So if you look on the margin right here, that the little bit of tissue that's kind of hanging off right there, we call that appendiculate when it hangs off, and it used to have a veil, right? So that kind of tells you that this mushroom had a veil at one point in time. So that's the only pleurotus that has an annulus or a ring in its initial stages. Okay, so identifying mushrooms with pink spores. Pink spores, as I mentioned earlier, they'll be true salmon pink in coloration, right? You'll get some that are light pink, and that's more in the coloration of pale spores, right? So you don't want to clump those together with true pink spores. And then there's two families, the Plutoceae and then the Entelomaceae. Those are the difference between the two is the gill attachment, kind of like how I pointed out earlier, right? So with Plutoceae, the gill attachment is free. That means that it's not connected to the stem. And entelomas, they have an attached stem, right? So the gills would just be attached to the stem basically by a notch or, various different kind of attached morphological characteristics. So Pluteus is this one right here. It's a saprobic genus. Not sure if this one's a really good example. Can you zoom in on the one on the left? Yep. So I'm trying to show you guys what I mean by free. Okay, so this is the outer layer of the gills right here. You see it's all pink, and then there's a little bit of ring zone right before the stem appears in the middle. And that's kind of what we would use to describe a free gill attachment, where the gills kind of stop short before it even reaches the stem. And as I mentioned earlier, the spore colorations is important, but as you begin to recognize mushrooms, you can kind of tell by looking at the mushroom and surmise what spore coloration it may be, right? Because when we're looking at this one and we're like, that one's kind of pink in color, it might have pink spores, right? So that's something that you can somewhat deduce without having to do a spore print. And if you're in a jam for time, you can grab, most of the time this works, you can grab like a Q-tip and kind of brush it against the gill face and just get like a little sneak a peek at the spore coloration from that. Okay, so Pluteus is a saprobic genus growing on dead wood, right? Sometimes you'll find it just growing out of the grass because there's buried dead wood in the soil, and that's what causes the confusion. We have two species that are really common here in North Texas. The Pluteus cervinus is what's commonly called as the deer mushroom because it's found in the woods. Deers love to eat it. Or deer's a word. Deer. Okay. So and basically there's no toxic Pluteus. There's some of them they could contain psilocybin, yeah, sorry. And those are none of those species are found in North Texas. So this one on the left is cervinus, right? And then Pluteus pedicellus is kind of famous for having these areolae cracked features on the center area of the cap, right? So as the specimen matures, it starts to develop these kind of cracked features right there, which cervinus doesn't have. So that's kind of how you separate those two. And then Enteloma is another mycorrhizal genus that contains a lot of species. So these are somewhat uncommon in North Texas. And whenever I find one, it's always really, really a pleasant surprise. But usually, as you can kind of see, can you zoom into one of those? These stems, they don't have that free ring zone. They're directly attached to the stem right there. And you see there's kind of just running right into the apex of the stipe. And the difference between Enteloma and Pluteus are the gill attachment, right? So if you have salmon pink spores, you want to look at the gill attachment before you cut off the cap and make sure you take a picture of it because you want to have it for record in case you want it to identified by someone on Facebook or anywhere else. And then for these entelomas, they always grow next to a certain type of tree. So, I mean, around this area, usually we have mostly hardwoods, but it would be important to kind of make a note if there's like a cedar or a juniper, we do get those as well. Oh, and I forgot, sometimes you will see a twist in the stem, which I don't really see here. And, you know, it's been argued that it's not really a characteristic, but a lot of times I have seen it and it is pretty consistent. But as the specimen matures, it can lose that feature. So just kind of be on the lookout for a twist in the stipe. So this is the brown spores, right? So once you cut off of the cap of a mushroom and place it on some foil, you get a brown coloration. And that's not to be confused with this pink coloration, right? So you can notice it's kind of got a salmon pink coloration here. And this one's a little bit more, it's kind of brown. Like, I don't really want to say it, but like crap. But anyway, so these mushrooms are, so a lot of these are smaller in stature and they're affectionately named LBMs or little brown mushrooms because people can't identify them. Yeah, and usually they're very abundant and they can be very difficult at times. That's why they have that affectionate name. And so to dive in, we're going to start with Anosabe, which is a mycorrhizal genus kind of similar to Clitosabe in the fact that it has the toxin muscarine. And these are also right up there with Clitosabe and the amount of toxin cases reported by digestion of animals in North America. They grow right next to oaks in North Texas, and you know, chances are you probably find one growing outside of your home. They're usually recognized by having a fibrillose or scaly cap. The fibrils are usually pressed downwards, and the cap will also split radially. If you can zoom in on this one right here, yeah, left one's a good. Okay, so if you see, it's kind of shooting out lines from the middle of the cap towards the margin, right? And then usually in Anosabes, they also have that little umbonnet feature right there in the middle of the cap. The umbo is what it's called. And at times they'll have what's called a prunose stipe. That means they have little white speckled dots at the apex right here, but you really don't see that here. They usually have medium to decently large fruit bodies. And the basal mycelium is usually always present and usually tends to be white. And sometimes they'll have a small bulb at the base. So as I mentioned, you don't want your dogs around these because they do like to eat them. Agrosabe is commonly encountered as well, decomposes wood, and it's usually found a lot of times, it's quite often found on mulch piles, right? So if you got mulch outside of your home or outside of your work or wherever in the springtime, especially if you have like, you'll see a tree planted and then they'll put mulch around that base of the tree, right? And then I've seen this happen a lot of times where these just love to pop up right out and have a little party, you know, right there by the base of the tree. And usually they're small to medium in size. The cap is always dry and it usually starts to crack like dried mud, which maybe can you zoom in on the left one? So if you can see right here in the middle, it's kind of cracking up like you would expect a place that hasn't gotten rain in like 10 years to crack. So and at times, Agrosabe can have an annulus, and then that annulus will usually fall off. But as you can see, the specimens on the right, they don't really have an annulus. So that's the ring around the stem. And then they usually are confused with Pholiota and Gymnopolis, which I will kind of cover here in a little bit. So Conosabe, usually smaller in stature. They're a little bit fragile. The stem is usually slender. As you can see, this one on the left is having a hard time standing up. And this one on the left is called Conosabe deliquescens because the cap deliquesces, right? As it matures in age, it can't support itself and it kind of just falls over and just says, I've had enough. So, and this will, you'll see this in the springtime here in the next couple weeks. As soon as we hopefully get some rain, this will start popping up. And as these are all brown sport in nature, of course, this will be kind of difficult to get a spore print from. But, you know, even if you cut off a cap in this state and you kind of put it on something, it might give you an incorrect reading because whenever the cap kind of decreases and turns to goo or liquid, it might leave some of that coloration from the cap on the paper or foil itself, and it might fool you into thinking that it's a different color. So you don't really want to sport print something like this. And then Konosuba also have a striated cap. That means it has lines coming down the top to the middle right there. And those are telltale characteristics as well. They usually get confused with Scytherella, which is another genus I'll get covered here in a little bit. So Pholiota, this genus, it can grow on a variety of different substrates, but they tend to be on wood, and it's saprobic. But I found this one on the left here in New Mexico, and it was after a wildfire had ravaged the area. So this one's a pyrophilous fungi, which means it occurs specifically on burnt material. And that's really cool because we don't really get that many wildfires in North Texas. I think there was one recently that happened. It's called like Eastland Complex. Yeah, you guys heard of that? So I'm not really expecting these to pop up after that, but you never know. You know, there might be some cool pyrophilous fungi that we haven't encountered that, yeah, you know, it could bring here. So quite often specimens will have scales on the cap and on the stem here. And that feature is usually ubiquitous. The scales can disappear with age. So, or if it like has a bunch of rain, you know, or environmental conditions can make the scales disappear. And typically they usually will have a ring or an annulus. But as you can see here, this one doesn't. So this is what kind of leads to some confusion. So rusty spores, this is one of my favorite genus. Just until like 2 or 3 weeks ago, this genus is called Cortinarius. They're famous for having rusty spores. And for the last 100 years, we've been calling all of these Cortinarius. But just 2 or 3 weeks ago, they split it into 10 different genera. So if it didn't get as much confusing as it was, you know, it's just got a little bit more confusing now. So, you know, it's still accepted to call them Cortinarius. We'll still know what you're talking about, because I think it's going to take a lot of time for people to catch up to understanding this split, because it's such a massive split. This was the largest genus of agaric fungi, which means gilled mushrooms, and it contained over 2,000 species, and it was just split into 10 different genres. So, you know, for the past 200 years, it's been working, but, you know, kind of they decided that based on certain morphological features, like I'll just give you guys a glimpse. This one here is a phlegmaceum because it has a bulbous base, right? And they tend to be slimy in nature. And this one here, it doesn't have a bulbous base, and it's kind of slender, and it's not really slimy. So based on these morphological features, this one's in a subgenus called Leprocype. They've actually made that subgenus into an actual genus. So I don't think Leprocype is one of the genuses that they came out with, but they made it into another genus entirely. So if there's morphological features to where we can kind of differentiate their just by looking at it, that's kind of what mycologists want to go off of to give them a description. So what you see right here is called the cortina. And also that's called an ephemeral veil. It disappears as a specimen matures, or sometimes it can be there, as this one is, throughout its entirety. And those are all spores that you see kind of stuck to the threads right there. And all Cortinarius kind of start off and have this feature. But in some species, it's not as pronounced, right? So that's a good indicator on identifying the species as well. So the presence of slime is important, whether it's on the cap or the stem or the entire fruit body, or if it's dry, if it's entirely dry. So if you pick one up and it's entirely slimy, then you want to make sure to note that. And because usually we can't tell that just by looking at it in a photo, unless it's just dripping with slime. Um, and then, so this one right here is a deadly species. I haven't seen it in North Texas yet, but it contains a toxin called oil-9. And that one, it starts to destroy your liver. So you don't really want to eat that. Gymnopolis is another saprobic genus, which can be confused with the brown spore genres. So these previous ones are all brown spore. That's the brown spore coloration, right? And then moving into the rusty spores, can you zoom in on that spore print there? So this can kind of be confusing when you're just getting into it, right? Because to me, that kind of looks brown, you know, but yeah, and the slight variations of color will take a lot of time to understand and recognize. And that's because something I kind of wanted to point out. And so this one is Gymnopolis, which has heavy orange spore prints, right? So this is known for being one of the most prolific spore producers in the fungi kingdom when it comes to agaric fungi, which are the gills. And I've seen it just, you put a cap on something and it just pretty much drops all the spores and you can't even recognize where the gills are anymore right there, right? Because that's how abundant the spores are. And Gymnopolis is known for occurring on wood, but it can be found growing on buried wood as well. So a lot of these species that do occur on dead wood can be found growing terrestrially on buried wood. So that's where the confusion kind of comes in. It's poorly understood genus with about 200 species worldwide and can be very difficult to get to species. A lot of Gymnopolis will have an annulus or a ring around the stipe, and usually it disappears with age, but it's not a ubiquitous characteristic. It is not 100% with this genus. And some species are known to contain psilocybin, which is the compound known for causing hallucinations. They're very bitter, and you know, I really wouldn't suggest people eating these to do that. So identifying mushrooms with dark spores. So these are the varying shades of dark colorations when it comes to spores, starting from agaricus, which has somewhat of a chocolate brown coloration to it. And then you're going into further dark as you kind of go into stropharia. And then Cytharella and psilocybe kind of had similar colorations. And then when you get to the ink caps, it's just like a blob of ink. At certain times, certain species will deliquesce and actually turn to goo. And that's where they got their name from, is because hundreds of years ago, people used them as actually writing, you know, for ink. So they're all saprobic. There is no mycorrhizal dark spore genus, and they're all found growing on the ground. So like I said, it can be kind of confusing. So you might have to refer to this chart quite often. Yes, sir. So a member asked if we have ink caps in North Texas. We tend to get a lot of them more in the spring and fall. I have one over there. It pretty much turned to ink, but then it dried in my little box right there. So when I lifted it up, it's very fragile. So when you guys do touch it, please be very careful. But they can be very difficult because they'll start to turn to goo, and then you want to preserve that goo. So yeah, it can be confusing, to say the least. The genus Agaricus is commonly associated with the button mushrooms or Agaricus bisporus, which are purchased at your local grocery store, right? You go and get your little portobellos. So a lot of these, Agaricus is the only genus that I know of that you can differentiate edibility based on odor, right? And I wouldn't suggest this for everybody because I'm sure some of you probably don't have the best odor identifying skills, but the toxic species, they have phenolic or chemical unpleasant odor. Some even smell like urine. So a good little trick to check for if you don't really smell things that well is to crush the base and throw it in the microwave for like 15 seconds or 10-15 seconds, and that'll amplify the smell, right? So but you want to make sure it doesn't smell like pee first before you throw it in your microwave and amplify that smell, because you probably it'd be difficult to get that out. So So agaricus can be confusing because earlier I mentioned where you can look at a mushroom and kindly deduce the spore coloration. Well, agaricus at first they have a pink color, right? So that's where you might get confused because in the younger specimens, they tend to have a beautiful pink coloration. And then depending on even if they sporulated or not, if they reach that stage, they can kind of remain pink throughout its life cycle, which kind of remains a little bit more confusing. But for a majority of specimens, as they grow older, they turn chocolate, dark black almost in coloration, as you see in this specimen right here. And they usually always have an annulus, and they have what's called a cobweb veil, which when it opens up, it doesn't really leave a skirt. It just kind of opens up with the margin and kind of leaves a little bit of the velar material on the margin surfaces. So you'll probably be able to tell by looking at it that it has an annulus by looking at the margin. So these species are usually entirely white, and in their button stages, it can be kind of difficult to identify. You can confuse them with other genres. And then also you want to, there's two different colors that agaricus brews, red and yellow. I haven't seen any of the red ones in North Texas. There's more of a wooded agaricus feature. But in North Texas, we do get ones that bruise yellow, and that's an important characteristic as well in agaricus. For the most part, the toxic ones that have an unpleasant odor will also bruise yellow. So those are important characters to check for. Stropharia is kind of uncommon up here, but I decided to cover it anyway because we might encounter it in the spring. I usually find some in the spring. They usually have a purple tone to the spore print, as you see right here. But as do psilocybe, Cytharella, and others, and that's where identifying the mushrooms with the dark spore prints can be kind of tricky and confusing. So Stropharia are usually viscid or slimy, and the cap will have an annulus that at times can dissipate. So if you see right here, there's the annulus. This is a Stropharia rugoso-annulata. It's the king Stropharia. This is one of the edible ones that is marketed at gourmet mushroom shops. And so for the most part, they're found on soil, but occasionally can be found growing on wood or dung. So psilocybe and Panalilius. So psilocybe is the most famous for being a genus associated with hallucinogenic mushrooms. And in Texas, the only psilocybe that we have is Psilocybe cubensis. The epithet cubensis, or the last part, the ensis, refers to a specific region, right? So in this terminology, they're referring to the region that surrounds Cuba. Well, it doesn't really make sense because they also find this in India. They call it Psilocybe cubensis. So you know, take that for what it's worth. So whenever you see that last part of ensis in the name, that refers to a specific region that mushroom is associated with, which goes with the location that I kind of mentioned earlier that you have to be aware of. The fruit body is known for discoloring blue upon handling or when broken or sliced. And the ones that we have here in Texas is found growing on dung, on cow dung. So they usually have an annulus, and it usually disappears as it ages, but it'll leave the deposit of spores on what's called the ring zone. So when you look at can you zoom in right here, Ben? When you kind of look at the middle of the stem right here, it has dark purplish spores left over. After the mushroom opens up, cap releases the spores, and the spores catch on to the ring zone, and in that instance, you don't really need a spore print because you can kind of tell by looking at it. Okay, so Panalilius is a species that typically do not have an annulus. They don't bruise blue, and for the most part, they don't bruise blue. And the stipe is slender and tends to snap in half like a twig. That's a really good characteristic to check for in Panalilius. You want to take the stem and just kind of break it like a small little twig. So Panalilius also has several species that contain psilocybin or the alkaloid that causes hallucinations. The only species that bruises blue is Panalilius cyanescens, and that's found growing on dung as well. So mainly that's more of southern Texas thing is where you find a lot more of these. For some reason, I've been to a lot of cow fields. Don't ask me why. Yeah, I haven't seen this, and I've only wanted to look for if I could locate this within the Metroplex. And so far, after years of looking, I haven't found it within the Metroplex of all the cow fields that I've been to. And it's weird because it seems like it skips over us and goes to Grayson County, and they have it up there in some parts of Oklahoma, but I'm not really sure what the deal is there. Cytharella and Deconica. I'm gonna start with Deconica first because I was just talking about in the cow fields. If you're ever in the cow fields, you'll run into this little specimen or this species right here. Deconica, they typically don't have an annulus, and in Texas, they're found growing on dung. The species are hard to tell apart macroscopically, but in Texas, the accepted name is Deconica coprophylla, right? Coprophyllus means that it grows specifically on dung. So coprophylla means that this is a mushroom that grows on dung. And the difference between that one, it has kind of an attached stem. You can't really see it here. It's kind of a horrible picture to show that one, but there's typically smaller in size, probably get about a few inches tall at max. And then Cytharella is more abundant and it's really commonly found all across the world. They're kind of known as the brittle stems, unlike Panolius, which I just covered, that had the feature to snap it in half like a twig. These are very brittle, so they're very fragile. As soon as you kind of pick one, it starts falling apart. And for the most part, for the dark sport genre, they don't really have that brittle stem feature. So when you're with the dark spores, that's something to check for. And then I don't know of any species in North Texas that have an annulus. There are some elsewhere, I think on the West Coast. So that's not something that you guys need to be concerned about. Okay, so coprinoids are the ink caps, and it's a term used to describe several genera that makes up the ink caps. A lot of these, not a lot, but a certain amount of these species, they'll deliquesce into liquid goo. And like hundreds of years ago, they used that as a form of ink to write on paper. And so four of the main genres in this genus or in for that term is Copernus, Coprinopsis, Coprinellus, and Parasola. So these can kind of be difficult to tell apart, especially if you're just starting out. And the key is to look for it. Can you zoom in right here, Ben? It's called velar material, which is the material that starts off whenever a mushroom it has a veil initially, and whenever it pops out of that veil, what's left over on the cap is the velar material, right? So these small little scales that you see there, those are really the determining factor in identifying which one of these genus, if it's present or not, right? So Parasola, for the most part, does not have any velum. So if you kind of scroll, sorry. Oops, sorry. So Parasola doesn't have any velum, right? And it usually, Parasola doesn't deliquesce in the goo, so it's not really a true ink cap, but it's kind of grouped in that category based upon the spore coloration, and its morphological features are quite similar. So let's see, Coprinellus is really common as well. Coprinellus mycoceus is a species that is commonly found growing in the spring, and they'll just be growing in your backyard, on your way, walking on the trail or something. And they're very common. And Copernus is typically a little bit larger in size, and they're most well known for deliquescing into goo, as you can kind of see if we zoom in right there. I think the edge face right there is kind of almost even dripping, as you can tell. And you can see this feature sometimes whenever you encounter this species. So that's Copernus right there. And that one's known for the shaggy mane. I don't know if anyone's heard that terminology. Copernus comatus is a species within Copernus, and that's called the shaggy mane. So moving on to the false gills, and these are the chanterelles, right? They're species within the two genera, Cantharellus and Craterellus. And they're called false gills because they don't really have the true gills. To me, still they kind of look like gills. You know, I'm sure to you guys they kind of look like gills, but mycologists don't call them true gills. They call them false gills because of the fact that they have interconnected webbings. When you see in between the gill faces, they have kind of like a vein formation that connects them together, and this is kind of like the ridges and the vein features that I kind of mentioned earlier. Sometimes with certain chanterelles, you won't even have really defined ridges or veins, right? There's a species called lateratus, and that one's called the smooth chanterelle because it doesn't really have a gill face. So it can be kind of confusing. But the main difference between Cantharellus and Craterellus is Craterellus has a crater.
Makes sense.
Okay, so that's kind of easy to remember there, right? So it's cratered in the middle. And in both of these genres, none of them are toxic, right? So they have a lot of edible species, a lot of prized species. They're mycorrhizal. Some people are contesting this, but I always just find them right next to certain trees. And, you know, so usually in the springtime and in the fall, we'll see a bunch of chanterelles. And I'm hoping to find some here in the next month or two whenever we do get some more rain on our forays. So the one thing about chanterelles is the spore printing them could be helpful because they range from colorations to from pale to somewhat salmon pink, you know, and it's really weird because I don't really know any other genre that kind of blends in white with the salmon pink, right? So, and also if you're trying to get to species, I might be able to narrow it down because having specific colors kind of tells you which one you would potentially look at. So notably, as I said, the crater formation in the middle of the cap for Craterellus is very important. It is long thought to be mycorrhizal, and the black trumpet is this one right here. I guess for having the cratered indentation in the middle. So I found some, you know, you look at this picture and to me, it looks like they're huge, right? They kind of look like, you know, like 5 or 6. But then when I found this in person, I was like, it's smaller than my pinky. And I was like, what is this? You know, like people like, you know, I found like photos online of people that had huge ones. So I'm like, I guess everything's not bigger in Texas. And then the Bolita CA. So two of the main ones that we have here in North Texas, and mostly they pop up in the fall. And I found these right next to each other, and they really prefer the same host tree because they're mycorrhizal in nature. They prefer live oaks. So basically what I kind of want to start off with is if you can zoom into this one right here. Like if you guys remember the word reticulum, the reticulate feature that I was talking about earlier is like this fishnet pattern that you see on the site right here, right? So it starts to get bunched in and closer, smaller formations here, and it's longer, more well-defined spaces here. And then you look at this one, it doesn't really have that formation. It's kind of hard to tell because it's a little bit smaller, but these have really defined lines, right? And this one doesn't really have that. So these two are different species. And I found them growing right next to each other, and they both bruise blue upon handling, and they both grow under the same host. So it could be very confusing. But this one right here is Pulchro Boletus rubricintrinus. And it's a mouthful to say the least, but it's a little bit smaller in size, and it tends to have more reddish colorations, especially towards the base right here. They usually always have a little bit of red deposit. Yes, ma'am.
Would you say that anything that bruises?
No. So the bruising blue, members asked if everything that bruises blue is edible. The bruising is just because it's being oxidized with being exposed to air. So whenever you kind of hit it with your finger, you're
activating the cells inside the mushroom to oxidize with the air. And that's just a notable characteristic to include in your description. Or if you take a photo, we could see it in the photo, right? But bruising of any coloration is not a sign for edibility. The general rule for boletes was that if it has yellow pores and the bruise is blue, then you kind of have to look into it further. But in North Texas, I haven't really found any of the toxic ones. You want to stay away from the red pores, those that have red pores. So I've only found one here in North Texas that had red pores, and then to this day I don't know what it was. But for the most part, in North Texas, we don't have any toxic Boleta. And actually, these are actually really good edibles. So if you'll find these more often in the fall. And this one right here is Boletus liridalis, the one that has the reticulum or the fishnet pattern I was describing. It usually discolors brown as well. So not the specimen itself, but the actual fishnet pattern turns brown as it matures from the spores that are being dropped. So basically, the fishnet pattern is how you tell these two species apart, the size, and the cap of liridalis tends to be more brown in coloration. So, and it tends to have some cracks in it. So here is this bag. I went to the post office. Do you have a question? Oh, sorry. I went to the post office and I stumbled upon a bunch of these mushrooms and I was like, oh my God, I just need a bag. So I went and grabbed like a bag out of my trunk and I started throwing mushrooms in it. And then this lady came outside and she's like, what are you doing? And I'm like, I'm just picking the mushrooms. She gave me a weird look and went back inside. So I gave plenty of weird looks. So basically, when you're looking at this, it's kind of hard to tell, but okay, I could kind of point them out to you. Ben, if you could follow my cursor. So right here, you see the brown reticulate pattern on the stipe. The cap is more brown colored. This one here has cracks on it and is more brown colored as compared to this bigger one right here is truly red in coloration on the cap. And it kind of stays red throughout its maturity as compared to the liridalis right here. So both of these are really good edibles. You'll find them more often in the fall right next to live oaks.
Did you read those?
I just say both.
Yeah, actually, I
Specifically.
Oh, you.
yes, sir.
So one of my friends told me you have to peel off the spore bearing surface. Is that correct?
No, you don't have to. Like I've had some that had bugs laying eggs in the pore tubes, so that's something that you always want to be cognizant about. Unless you want some more protein, why not just throw it in? So let's see. Okay, let's see. Suillus is a large genus which is primarily associated with pine or coniferous trees. They're known for having these glandular dots on the stem, right? So these dots are very typical of Suillus and is a very good defining characteristic of this genus. They also tend to have larger pore sizes, and the specimens will have an annulus at times that can disappear. Quite often, you'll have to look at the margin and see if there has any bit of a velar material left over. But if you can see right here, it's barely starting. Can you zoom in on the bottom one? It's barely starting to lose the ring or, well, the ephemeral zone of the veil. And if you see the pore sizes, they're a lot larger with this specimen here. And that's really characteristic of Suillus. So orange to yellow to brown colorations found right next to pine. Okay, so a lot of these, you'll find it right next to pine. And Strobilomyces, aka the old man of the woods, we'll see a lot of these here in the spring. They'll start popping up, and I was warned about this before I ate it, and I really wish I wouldn't have. But I don't know, some people they really like it. And this was the one that had the bugs in the tubes, and ever since I kind of found those, it's been hard to burn that out of my memory. And then every time I'm like eating them, I'm like, think I'm eating like larvae from some weird insect. So anyway, Strobilomyces is more commonly known as the old man of the woods for its namesake species, Strobilomyces strobiluceus. In North Texas, we get all three of these species: Strobiluceus, Dryophilus, and Confucius. And Confucius, not to say the least, because it is confusing to separate these apart from each other. The Dryophilus will typically have a little bit of a lighter coloration, as you see in this specimen here. All of these will bruise red when you slice them, and they kind of have a gnarly coloration too, as you see right here. And they have a veil, and they have velar remnants around the margin of the cap right there. And usually Confucius is the species that has a little bit more of erect scales, right? So the scales are kind of popping up as opposed to the other ones, but it's still very difficult to tell them apart macroscopically. So Lexinum and Tylopilus are two genres that we don't really get that much of in North Texas, but I have seen Tylopilus in the spring. So I just decided to cover them anyway. And Lexinum, they have distinct scabers on the stem. Can you zoom into the left one? Thank you. So you see these black scales on the stem. They're really a noticeable feature throughout this entire genus, right? So whenever you see this, you can pretty much bet it's a Lexinum. And then a lot of these species, they will bruise a certain coloration when you cut them in half. So you want to do a cross section as well. And then Tylopilus, they will have a lot of species that are bitter, right? And doing a taste test is really going to help you get the species in Tylopilus, because some of the species are bitter and some are not, and they really look the same. So the ones that are bitter is one species, and the one that's not is another species. So that's how you get to the species there. So now to the polypores. These are called polypores because they have pores, and there's a lot of them. So poly means many, and pores, right? So there's a lot of pores. Pretty easy right there. They're primarily saprobic and decompose dead wood. Some polypores are pathogenic or parasitic in nature and can infect the host, typically through the roots, and in some cases cause disease and or death to the tree. So for people that kind of get like an infection in their tree and they don't really know what's going on, the tree starts to die, it could be a fungal pathogenic infection causing a disease in the tree and eventually leading to its death. So there's two types of polypores. Those are stipitate, or those that have a stem, and those without a stem. Those are the brackets, right? They're just fused to the wood. In the North Texas, we find polypores throughout the year. So in the off season, that's what we go out to find, our polypores. So polyporus, I put lentinus in parentheses because this is another confusing part where a lot of the species from polyporus were recently moved into lentinus because of DNA sequencing. So all three of these species are now in lentinus, but used to be in polyporus. And lentinus is a genus with gills, right? So that's where it's even more confusing. So that's the only polypores in the lentinus genus. It's really confusing, and I get a headache every time I think about it. So the most polyporous species will be stipitate or have a stem. And the most common one that I actually seen posted in the Texas Mushroom Identification Group on Facebook is this Arcularius. And that really tells me that that's the start of spring, right? So we officially have spring started because the mushroom just told me that. And it's only found in the springtime. So Tricholoma, lentinus tricholoma is very similar to Arcularius, and the fact that the morphological features are the same, except this has long eyelashes on the margin of the cap, right? So it's really cool to find this in person. And then the brumalis has smaller pores, and it's just got a browner coloration, so it was a little bit easier to tell apart. So Ganoderma, some of you may have seen this out on our forays. We usually find these almost every foray. Ganoderma are among the most commonly known and recognized polypores. They're also known as reishi, traditionally regarded to have a lot of medicinal properties. Chinese have been using these for thousands of years. And younger, fresher specimens that have what is known as artistic pores, or a pore surface that can easily bruise once pressure is applied. Check out this picture right here. It's pretty cool, right? So this is called the artist bracket, Ganoderma apollanatum. And that's because the pore surface bruises so easily that you can really draw on it, and it's really cool. So Curtisii is another common species that we have here, and that's the one with the stem. but at times, as you can see, this one doesn't even have a stem right above it, so it could be very variable. And then sisal is this one that's fixated exactly on the tree itself. And sisal just means it's kind of stuck in place or fixed to the tree. So all of these have medicinal properties. People make them into tinctures and take a few drops a day. Trametes as well for this namesake species right there, Trametes versicolor, also known as turkey tails. So this one is highly regarded as anti-cancerous and anti-tumor properties. And it has the species name versicolor is applied because it has a lot of variation in color, right? So you can really see it in all sorts of beautiful colorations. And then also the one more that I have right here is Trametes sanguineus. And we have two polypores that are orange up in North Texas, and I have both of them here. Sanguineus is separable by being, it's kind of got like a disc shape to it, and it's really thin, as opposed to cinnabarina, which is just kind of a big mass of a clump onto the tree. And sanguineus usually has a stem attached at the base right there, somewhat stipitate. Okay, and then the lactinia and esculi both look like this, right, from the exterior. So usually you have to flip them over to get to species because you have to look at this pore surface right here. So this is the difference between asculi, which if you can zoom in there, thank you, sir. I'll try to get a little bit closer. So you see this one has like lines through it, kind of going through it, has like these weird lines. Instead, this one just has circular pores kind of. So and these lines are, when you actually see these in person, these spores, the pore size for lactinia is a lot smaller than it is right here. This is just amplified to see it on the screen. So the pore size for lactinia is smaller and asculi is a lot bigger. Okay, so Laetoporus is the chicken of the woods. And, you know, this is called chicken of the woods because it tastes like chicken. It has the texture of chicken. And it's a really cool find every time I've seen it. And it's very uncommon, unfortunately. I wish it was a little bit more common. But they're usually brightly colored. And in North Texas, we get two main species, sulphureus and cincinnatus. Sulphureus is recognized by having this yellow sulfur color on its underside for its pore surface, right? So that's how it got its species name right there. And cincinnatus is recognized by having its white pore coloration. So if you see right here, this one on the right here is sulphureus, and this one on the left is cincinnatus. So this one has yellow pores, this one has white pores. And that's how you tell the difference between those two species. They're both edible and they're really delicious. So Anotis and Felinus, the coloration of the pore surface is important. Like I mentioned earlier, if you ever feel confused that you don't have a polypore, break it, right? You want to break the tubes, and then you'll see this kind of, I hate to keep asking. Yeah. So if you see, this is the pore surface right here. These are the tubes going up and down, right? So the tubes are, in this species, are very long, right? So when you break it in half, you see these distinct tubes, and it kind of tells you it's a polypore. Okay, so let's see. And then the Anotis, like Ganoderma, have artistic pores, but they're not as artistic in print, in quotations. They do bruise brown, but not as significant. Some have a softer texture, and some are really they have this tomentose, which is a fuzzy type of characteristic on the cap, and that's what really tells it apart from Felinus here, which has this cracked cap, which almost looks like the bark of a tree, right? It almost looks like it's fused into the tree. And I have a specimen there to kind of show you guys. And so Fomus is another genus of polypores, and this is kind of like the hoof fungus, right? So this is called the hoof fungus because it looks like a hoof. We really don't get this in North Texas, but we do get a lot of Fomus fasciatus, which I have another species right there. And it's quite often it is confused with Ganoderma apollinatum, the one that is the artist bracket. It does bruise, but not as significant on the pore surface. So the pore surface here is brown in mature specimens, and it can be white in younger specimens. So it kind of gives you the idea of the spore color. And now to the crusts. The crusts are another loose terminology applied to many different genera and species. These are what's found growing on dead wood, and they grow flat on wood, right? Sometimes they'll develop somewhat of a cap, but they abort, and that's called effuso-reflexed when it's almost a cap, but it's not. And then some features you have to look for is if does it have pores, teeth, bumps, or does it have like a smooth surface, kind of right here. This one's called Piniophora albobatia, but it's also called giraffe spots because of its similarity, look similar to giraffe spots. So Urpex lacteus has spine-like teeth formations coming down, and that's another really common one that we find here. And then Morchella, aka the morels, these are all hollow on the interior, right? So whenever you split one in half, they should always be hollow on the interior. They're notoriously difficult to identify two species. You mainly want to look at the pits and the ridges. You want to look at the size of the pits. You want to see how many there are. If this one's really deep, it has deep pits compared to this one here. And usually they're mycorrhizal, but people have been disputing this for the past, ever since they found these morels. So these are false morels, the Gyrometra and Verpa. So unlike the true morels, the Gyrometras, they don't have a hollow interior. They kind of have like a solid mass that when you split it in half, it's just kind of like a cauliflower or something like that. And Verpa is very similar to morels, but the cap hangs off like a skirt. Okay, so the main identifying factor is the cap with Morchella is attached to the stem, right? And then you go to Verpa and it's basically hanging off like a skirt, so it's not attached to the stem. So, and then Verpa, it's commonly mistaken for Morchella. It is, I mean, some people say it's not edible, some people, edibility is always contested between different people, right? So what one person says is edible, another person can have adverse reactions. So Helvella is quite a unique genus in the fact that it has varying morphological features for the cap, but depending on the species, it also has a consistent looking stem throughout the entire genus. So if you look at this stem, it's really unique. It kind of looks like if you took all the skin off of your arm, right, and you just kind of had like Terminator style or something. It has like pits, and it is also hollow on the interior. But if you could recognize this stem, right, you could recognize this genus. And these are a couple of species I found in New Mexico. They do have the same stem, right, but the cap colorations is different. And this is the cup. This is one that's similar, Helvella acetabulum, and it has a cup formation instead of a cap, which is really cool. And they're all mycorrhizal and closely related to Morchella. So this is kind of a weird one. This is the stinkhorns. So this is a rare species that I found, I think last year or the year before that. It's called Lysurus species 01 because it hasn't even been named yet. And this is the spores right here in that gleba. It's a really sticky mass that insects are attracted to. And when the insects land on the gleba, it attaches to their body, and wherever they go, they spread the spores, and that's how their redistribution occurs. So a couple of different species that we have here that's quite common is Mutinus elegans and then Phallus rubicundus. As you can see here, they're very similar in their morphological features, except the main distinguishing characteristic here is the cap dissipates into the stem on Mutinus elegans, while the cap is kind of hanging loose on Phallus rubicundus, right? So this kind of dissipates into the stem and it's always attached, where rubicundus is just kind of hanging loose. So those are kind of some characteristics for, this is a jelly fungi. So Arcularia is very recognizable because it looks like an ear. Many other species, it will actually form together, and sometimes it doesn't really look like an ear, but quite often you will see this formation. We do have several different species here. They usually will dry up when they lose their jelly consistency, so they'll try to turn into almost a cracker dried state. And then some of the more common ones is Arcularia, as I mentioned right here. This one is Exidia, which can be mistaken for Arcularia quite often. they fuse together and they're kind of blobs, right? They're more blobbed out, and Arcularia is more of a defined features. And Tremella is also known as witch's butter, right? Tremella mesenterica. There's usually orange in coloration, and that's one's pretty recognizable. So coral fungi, usually we don't really get that many in North Texas, but in spring I have seen a bunch of these pop up. And one of the main, they're notoriously difficult to get to species. They usually tend to have same colorations. And one of the main genus that we have here is Ramaria and Clavelina. So the main identifying characteristics here is the tips of the corals, right? So you have to look at the tips. These ones kind of have like a spine formation, but over here is more of like a crowned projection for the tips. And then you also want to see how fused together they are. Are they clumped in together, or are they bunched out and spaced further apart? And then the bird nests, really cool, really recognizable. They really have a stature similar to a bird's nest with eggs inside, right? And those eggs are the spores. And whenever it rains, the raindrops hits the spores, and then the spores will distribute out around the surrounding area and restart their life cycle again. So two of the main species that we have here is Cyathus stercoreus and Crucibulum laeve. The main difference between these two is the tomentose or fuzzy exterior, right? So this one has the spinal projections coming out of the exterior, and this one doesn't, right? And then all these start off their life cycle. They have what's called a peridium. It's like this tissue layer on top of the eggs. And whenever it matures, that tissue layer starts to open up and reveals the eggs inside. And puffballs are really cool. We'll get a lot of those in spring as well. I kind of wanted to include this picture in here because quite often I will see this. It's just the sterile base left over after a mushroom has sporulated, right? So it blew off its top like a volcano, and it's just kind of left over with this sterile base. And the sterile base can be there for months on afterwards, right? So one of my favorite things to do was just go by and kick it, just kind of fly over. It's really, it doesn't have any weight to it, and it kind of just flies up, and it's really cool. And so basically the two main genre that we have here in North Texas is Lycoperdon, which usually has like these spinal formations. It's kind of like these gem-studded warts kind of coming out of the skin layer. And Calvatia, which usually doesn't have that feature, and they tend to be larger in size. And Calvatia, for the most part, we have some species here that have purple colorations to their spores. So that's a good identifying characteristic for Calvatia as well. And then truffles and earth balls, right? So truffles, subterranean fungi, right? They grow right next to the roots of certain trees. They grow off of the trees root system, and they're highly prized. They go for, some species can go for thousands of dollars per pound. And in Texas, we don't really have that many except for the pecan truffle, Tuber leone. And if anybody has access to a pecan orchard, we'd love to come pick it one day. Yes, ma'am. Yes, ma'am. Oh, you do? Cool. Yeah. Yeah. No, that'd be cool if you can like find one day and bring it in. We'd all love to see it. Oh, cool. Yeah, I remember it was just saying she has access to a pecan orchard, so that's really cool. You have to go there with like a rake and pretty much start raking stuff up to try to get lucky with these, because yeah, that's cool. One of the members says she has a truffle hunting dog. That's really cool. I have to borrow that one. Like one member just mentioned, you'll see people with dogs and sometimes pigs, but pigs will usually eat the truffles. That's why they're not really used that much. So Scleroderma is a genus that looks can be similar to puffballs. I mean, not puffballs, but truffles. Except they have this exterior that kind of has like a scaly, flaky surface to it, as opposed to the smooth exterior of a tuber or actual truffle. And then when you slice them in half, they have a purple coloration in the interior. So Scleroderma is toxic, right? And these are also one of the most reported cases of dogs being poisoned because they think it's like a ball or something. Yes, ma'am. Yes, ma'am. So they can range between purplish to jet black in coloration. And then sometimes in younger specimens, before they start the sporulation, they won't be purple or black at all. They kind of have like a tan coloration. So that's where it's kind of confusing. So, and then cup fungi, there are many different genera that fall under the category of cup fungi. And the basic requirement is just to have a resemblance to a cup, right? So as you can see down here, there's several different species I highlighted. Pizziza is a genus that we most commonly encounter, and they're a saprobic growing just on the ground. And Otidia is similar to Pizziza, except they cut off halfway, right? So instead of having the full cup, it's like a half cup, like split lengthwise. And then this one's Ernula craterium. And this one's really cool because there's two different kinds of cup fungi, and there's the stipitate ones, and then there's the ones without the stem, right? So I don't know if you guys can see this one, but that's the stem right there coming out for that, the specimens right there. And these don't have a stem. So just like polypores, there's two different kinds of cup fungi, the stipitate and the non-stipes. Club fungi. So these are the ones that kind of look like a club, pretty simple there. And pretty much there's two different species I highlighted here. They're kind of tough fruit bodies. They typically grow on the ground, but Xylaria polymorpha is a saprobic on wood. And that one's kind of known as the dead man's fingers because sometimes it pops out of the ground on buried wood and it just looks like it's coming out of the ground and it looks like fingers coming out of the ground. That's why it's named dead man's fingers. And so the earth stars, they kind of have, they're similar to puffballs, but they have an exterior layer that opens up like a star. So, when you squeeze them, the area in the middle is called an ostiole, and that's where the spores shoot out of, right? And they really have some gnarly looking spores, which I hope to bring in the future and we can look under the microscope. And then cordyceps and friends. These are some of my favorites. The entomopathogenic fungi, right? So these are these fungi specifically infect an insect in most cases to cause a disease and kill them. You know, sadly, the good thing this doesn't happen to humans because we'd kind of be screwed. So this one here is a moth that got consumed by Acanthomaeces aculeatus, and my buddy Logan found at the winter foray in Louisiana. And this one that we found at our last foray in Fort Worth, Bovaria bassiana, which I think is kind of more common, but look at the size here. That dime is there for a size scale, right? So if you're looking at, imagine a forest floor covered with leaves, and then you try to find this. So I think it's more common, but people are having difficulty spotting it. And these are my references. I'm sorry to kind of hold you guys a little bit over the allotted time. If anybody has any questions, I thank you all for being here. If anybody has any questions, please go ahead and send me an email. A couple of announcements I want to make really fast. We are going to be ending our spring foray sales, which is the weekend foray coming up in April in 1 week. So if anybody wants to kind of get their last minute tickets, please purchase them this week because it'll be your final week because we need to give a head count to the caterer to know how many people to prepare for. And then other than that, we have our April foray in Southlake coming up in mid-April. So there's event details on our website. I'm hoping for some more rain because this past foray was kind of really bad. It was the worst turnout for fungi we've had since we started doing these. And so I hope that trend kind of ends. I think there's some rain in the forecast on Monday. So yes, ma'am.
And also, if you're going on a trip, I send an email with your food preference or vegetarian. We really need to let us know because we need to order caterers.
Yeah. Yes, Jen. Yes, please make sure you do sign in. Thank you all for being here today. We hope to keep offering these classes in the future. I don't think we're gonna have one in April because of time constraints for. Yeah, but our next one will be in May. So I hope to see you all here. Thank you.
Yeah, you got it.
Hey, thank you so much. It's a pleasure having you. Thank you.
I have a question for you.
Sure. Oh, that was a good one. Edgeless player. Oh, where'd you find that at?
Oh, out of the state, where all the cool mushrooms are. It's somewhere rainy.
Sorry, man. This is to bring.
Hey, I didn't know. No.
I found it in the back. Oh, okay. Oh,
The one with the block.
yeah.
You want to go ask him if you wanted the block, if I get one. You got an extra one? You got an extra one?
Yeah, enjoy it, man. That's great. Oh, yeah.